Every year, more than 100,000 people are diagnosed with leptomeningeal cancer, and yet we do not have the tools to prevent, diagnose, or treat this disease. The Leptomeningeal Cancer Foundation is here to change that. Working with medical experts, researchers, patients, and their families, we will raise awareness, educate patients and healthcare providers, and support research. Please visit us at lmcancer.org and follow us on social media. We are here to support you and ensure that you never feel alone.
Good morning, everyone. Welcome to today's webinar. We're so grateful that you are taking the time out of your day to join us. I'm Helene Bader. I'm the founder and president of the Leptomeningeal Cancer Foundation, and today we are talking about clinical trials in leptomeningeal cancer. And this is such an important topic. As many of you know, I started this foundation in honor of my son Theo, who died at the age of 17 from leptomeningeal spread. He unfortunately did not have the opportunity to do a clinical trial, but I wish more than anything there had been one that would have worked for him. So it's really encouraging to see more and more trials, giving patients, like many of you here today, more opportunities. We have really great presentations today, and I'm really excited to dive right in. Just before I do, just a reminder that the information presented is not intended as medical advice or as a substitute for medical care. So please speak with your doctor about any healthcare decisions and your personal care plan. So first, I'd like to welcome Heather Davis, who has been graciously volunteering her expertise and time with the foundation. Heather has more than 30 years of experience in the clinical research and drug development industry. As Chief Operations Officer of an oncology-focused CRO, she ensured quality execution of all clinical trials. She's a passion for oncology clinical research, having had a personal experience with losing both her mother and grandmother to cancer. Heather will give a brief overview of clinical trials so we can all start out with some basic knowledge before diving into the specifics of the trials. Heather, I'll turn it over to you.
Thanks, Helene. Good afternoon, good morning to everyone. Today's brief overview of clinical trials is focused really more for those who don't have an awareness or understanding of clinical trials. And so I'll go through from here. Next slide, please. First, I'd like to talk a little bit about what clinical trials are. Clinical trials are research studies that are in oncology specifically. They're looking at new investigational drugs for people with cancer. And the purpose is really to determine whether the new treatment is safe and effective. People who participate do so as volunteers, so they must consent to participate in the study. And there are study-specific criteria for each trial that patients need to meet. And that can be anything from the type and stage of cancer to the current health status, their recent treatment history. And this is to allow us to look at a uniform patient population to help us to ensure safety and as well to help us be able to measure effectiveness of treatment and safety. Next slide, please. And why is clinical research important? As Helene said, for her son, she wishes that there was an option. Really, it helps us. There's three things really, and there may be others that you can think of, but it really helps us understand the disease better. It helps us to understand about diagnosis, potential prevention options, new treatments that we can develop, and really existing treatments, can we improve those? There's been an incredible amount of progress made in oncology clinical trials over the past years. We also, through the clinical trial process and the rigorous requirements, it ensures safety and effectiveness during that drug development process. And this last point, I think it's really important to understand that it may provide early access to potentially promising treatments, but there's no guarantee on a clinical trial that you'll receive the investigational drug. And there's no guarantee, because we don't know, if that drug is effective and if it is safe yet. Next slide, please. Phases of clinical research. There's actually 4 phases, but we'll focus on the 3 today. And the reason why I don't include the number of patients is because for different indications, some studies have a large number of patients, and some studies where patient populations are more rare, there's much smaller. There's also some trials too that go from a phase 1 to a phase 2 and then register to get that trial approved. And some trials go all the way through phase 3 for the regulatory pathway to get that trial approved. It just depends on the status that trial is provided. For phase 1 trials, those are where we're looking at the initial safety in humans. So this is the first time that we've introduced this investigational drug into humans. And we're looking at safety as well as determining the initial dose that we'd like to do. The initial dose and then as well the final phase 2 dose that we're looking at. During that time, we're also understanding how the body is processing the drug. In phase 2, we look at a larger number of patients and we more further evaluate effectiveness and further understand safety, and we start identifying what potential side effects may be. In phase 3, we go into an even larger number of patients, and at that point in time, we compare it to the standard treatment for that particular disease. We also confirm safety and efficacy, and then we determine if we've met the endpoints to be able to register for a new drug approval. Next slide, please. Informed consent in a clinical trial is critical. I was a research coordinator, so I worked with patients directly and with investigators and talked through consents. It's really important that we provide this clear information so it allows patients to understand what they may be getting into if they're involved in a clinical trial. It also ensures ethical standards. It ensures that they understand what their rights are and then what their involvement entails. What's really important for us too as researchers is that if a patient remains on a study for the entire trial, then we're able to understand essentially the outcome of that patient and how that drug potentially worked in that patient. And the best way that we can see patient retention is typically by helping them understand what they're volunteering for and what their commitment will be. However, it's really important to understand that you don't need to stay on a clinical trial. If you enroll in a trial, you sign consent, at some point in time, if you decide you'd like to withdraw from the trial, that's completely up to you. You can ask questions, you can withdraw at any time. You can even withdraw after a trial is done and ask that your data be removed prior to a certain point in time. Next slide, please. Question to consider as you're going through considering whether or not you'd like to participate in a trial. The process of consent is typically provided to you with the consent form, but then they're actually walking through these questions and answering any questions you may have. So a lot of these will already be covered in the consent form and in those discussions. But obviously the purpose and the phase, what are the risks and benefits? What will your time commitment entail? What type of investigational drug? Is it oral? Is it IV? How does it compare to treatments that you may receive if you don't participate in the trial? How will your safety be monitored? If the drug is working, this is always nice to know. Will you have access to it after the study ends? And next slide, please. This is safety monitoring. So on the trial, and you can ask your investigator, your doctor more about this as well. There's all sorts of levels of safety that we're looking at from a patient perspective to a study-wide perspective, from the initial development of the trial through the conduct of the study at many different levels. And so this is just a brief summary. And that's what I have for you today. Thank you.
Thank you so much, Heather. That was a perfect way to set the stage, and I just really appreciate having that knowledge before going into the details of the clinical trials. Okay, so now we're going to move into the specific trials, and just so you all know, we're going to take questions after each doctor's presentation, not waiting for the end this time, so please put your questions in the Q&A as they arise, and we will get to as many as possible. And if you sent one in with registration, we will include those as well. So next up is Dr. Jessica Wilcox. Dr. Wilcox is an assistant attending neuro-oncologist at Memorial Sloan Kettering Cancer Center in New York. She cares for patients with primary and secondary brain tumors and neurologic complications of cancer. She has a long-standing interest in emerging treatments for brain and leptomeningeal metastasis and has published several high-impact papers on the modern management of CNS metastases. She is the principal investigator on the trial she will share with you now. Welcome, Dr. Wilcox, and I will turn it over to you.
Hi, Helene. Thank you so much for that lovely introduction and for inviting me to speak to you all today. It is an honor. Just to give a very quick overview, leptomeningeal metastases represent the access of cancer to the cerebrospinal fluid. And when they get to the CSF, they exist in two states. They're either adherent onto the surface of the brain and the spinal cord, which you can see on an MRI, and they're also floating in the spinal fluid, which we use lumbar puncture analyses to test and measure. And normally, in the absence of cancer, the spinal fluid has a very low number of immune cells in it. But once cancer gets into that space, you can see here on this plot that there is a big increase in T cells and B cells and macrophages and all of these different immune system cells in green, blue, and red, in addition to the cancer cells here in yellow and orange. And when you compare the components of the spinal fluid compared to blood, the spinal fluid is very, very different. It's very low in oxygen, low in glucose and nutrients, and particularly low in iron, which is primarily what I'm going to talk to you today. Now, iron is a critical component for multiple cellular processes in the body for both cancer cells and normal cells. Iron is involved in DNA synthesis and repair. It's involved in energy formation. And we believe that iron is particularly important to the growth and survival of cancer cells in the spinal fluid. We see here that when cells are in a very low oxygen environment, we see a big upregulation in the machinery the cells need to use in order to process iron and get iron into the intracellular components. And when we look at the spinal fluid of patients with leptomeningeal metastases and without leptomeningeal metastases, we see a big increase in the iron concentration in the spinal fluid in the presence of cancer. Now, this is all really beautiful work that was done by Dr. Adrienne Barr from Memorial Sloan Kettering. She's the mastermind behind the concept of this study. And the short of it is that what her lab has determined is that iron is shared both by the macrophages, which are the immune cells in the spinal fluid, and the cancer cells in the spinal fluid. And the spinal fluid cancer cells will release these receptors to try to pull iron away from the macrophages and into their cells so that they can use that iron in order to grow and divide. And we were trying to come up with ways with which we can manipulate the iron concentration in the spinal fluid so that the cancer cells can't use it anymore. Now, deferoxamine is what we call an iron chelator. This is a drug that removes iron from the body. It's FDA approved for intravenous use for iron overloaded states, but has never been tested directly into the spinal fluid before. And when this is administered intravenously, we just don't think it gets to high enough concentrations into the spinal fluid to really have tremendous activity there. So what Dr. Barr's team did was they injected mice with leptomeningeal cancer cells directly into the spinal fluid, and then they started treating those mice with this drug, deferoxamine, also directly into the spinal fluid. And what they saw was when you put deferoxamine into the CSF, there was a big reduction in the concentration in iron in the spinal fluid. And what that resulted in was a reduction in the amount of cancer that was able to grow in the spinal fluid, and also prolongation of survival of the mice. So we took that data and turned this into a first-in-human phase 1 study of deferoxamine intrathecally in patients with leptomeningeal metastases. And so this is a study that I developed with Dr. Barr. She was the principal investigator of the phase 1A component, which I'm going to talk to you about today. And now I am the principal investigator of the phase 1B component, which is currently enrolling. So as Heather had just discussed with you, as a phase 1 study, the purpose of the study is to determine the safe dose and also the safety profile and toxicity associated with administering deferoxamine directly into the CSF. We're also looking at pharmacokinetic properties of the drug, so how well is it able to clear out of the spinal fluid. And in phase 1B, we're going to look a little bit more into how well it works for patients with leptomeningeal metastases. And the way that this drug is administered is that all patients will get an Ommaya reservoir, which is a little device that gets implanted into the ventricle space of the brain and allows us to access that device just to take spinal fluid out, to send for testing, and also to administer drug directly into that space. And so we're administering deferoxamine at a schedule of 2 times a week for 4 weeks, then once a week for 4 weeks, and then once every other week until we see cancer cell progression or intolerable toxicity. Now, we've completed the phase 1A dose escalation component of this study, which allowed us to determine what is the safe dose that does not cause any excess toxicity in patients. And now we're enrolling in the phase 1b component of the study, where we're taking that safe dose and testing it in 20 patients with leptomeningeal metastases from any solid tumor. And in order to enroll, some key eligibility criteria is that you need to be 18 years or older. You need to have what we call a Karnofsky performance status of 60 or greater, which basically means strong enough to be able to be, up and out of a chair for more than, a half of the day, and able to take care of most of your activities of daily living, like eating, bathing, using the bathroom. Again, cancer from any solid tumor is eligible as long as it's in the leptomeninges. Patients all have to have that Ommaya reservoir that I talked about. Patients cannot have what we call a VP shunt or VPS. And what that is a device that patients with high intracranial pressure receive to drain the spinal fluid into the belly. And the reason why we don't allow for the use of a VP shunt is because that could alter the pharmacokinetics and the drug durability within the CSF. And then moving forward, this is some preliminary data that was just presented at ASCO 2 weeks ago. This is looking at the first 8 patients that enrolled in our phase 1A. You can see here the average age of our patients was 50, but there was a wide range from 26 years old being the youngest and 69 being the oldest. There was a mix of primary malignancies. So we had 4 patients with breast cancer, 2 with lung, 1 with colon, and 1 with sarcoma. All patients that enrolled on the study had stable disease in the rest of their body. And 62% of the patients continued also on systemic cancer-directed therapy as well while they started this drug. And all patients had received prior lines of therapy directly to the leptomeninges. So that could include leptomeningeal-directed radiation, like whole brain radiation or proton CSI, or other intrathecals or other drugs designed to treat their LMD. We found that deferoxamine was very well tolerated and behaved in the body very similar to other drugs that are already FDA approved for intrathecal administration. We see here a number of grade 1 and 2 toxicities, which means that those toxicities were mild or moderate. And the most common thing we see was vomiting, nausea, some chills or body aches. A few patients had headache or fever, dizziness. 3 patients had what we call grade 3 toxicity, which means severe enough that they needed to be evaluated in a hospital setting. But all of these grade 3 toxicities were self-limited, and that included vomiting, a tremor, and transient loss of consciousness. And then this is just looking at how long patients continued onto the clinical trial. So in the y-axis, each one of these bars represents a patient, and on the x-axis, this is how many months they were on study. And in dark blue was how long they received the deferoxamine, and in light blue, after they stopped the deferoxamine, shows how long they survived. Now, we see a wide range of responses to the deferoxamine, but importantly, we had 3 patients that were receiving the drug for almost 5 months or longer, and 2 patients that continued with the drug for 1 year and over 2 years after enrollment, which is a really, really phenomenal response for this disease. And the median survival of patients enrolled on our protocol was 10 months. And so in conclusion, this drug appears to be safe. It seems to be well tolerated and effective in some patients with leptomeningeal metastases. Key things, if you're interested in enrollment, is that, again, you have to have any detectable leptomeningeal metastases. So that could either be abnormal enhancement on an MRI or abnormal CSF, normal intracranial pressure dynamics in Ommaya reservoir. And then if you're interested in hearing more about it or if interested in enrollment, you can shoot me an email at this number, at this email address below. You can also find the study on clinicaltrials.gov.
Thank you, Dr. Wilcox. That was incredibly interesting, really helpful. We do have a few questions. I'm actually going to ask them to you together because I think they're kind of connected. But one person did ask about would dietary iron or iron supplements make LMD progress faster? And then another person asked, my iron has been very low. Would this be because it is all going into my CSF, like a sign that my LMD is progressing? So can you address both of those?
Oh, sure. So, number one, I don't think that taking extra iron in your diet or by supplements would feed the leptomeninges. The iron level is regulated very tightly in the CSF. So even if you take, an excess amount of iron by mouth, it's not going to just automatically all feed into the CSF. Um, iron levels, to your second question, are often very commonly low in the blood of patients with cancer. That's for a lot of different reasons. A lot of patients are anemic or have low red blood cell levels, and that has to do both with the cancer, but also with some of the systemic therapies that they're receiving. So to that question, no, I don't think that having low iron in your blood means that it's all funneling to the spinal fluid either.
And then one other question, are only patients who have completed their brain radiation eligible for this?
Nope. Patients can enroll whether or not they've received brain radiation. The most important thing is just to be sure that we know that the intracranial pressure is normal, and that can be measured with a lumbar puncture.
But they don't have to, could they still be doing radiation or it has to be completed? Like they,
Oh, I see. So,
yeah,
they can enroll after 7 days have passed after radiation finished. So they can't get leptomeningeal directed radiation during the study, but they can start the drug very shortly after radiation has finished.
perfect. And then one more question, and then we'll move on to our next speaker. Is it possible to have my shunt removed that I had placed 10 years ago?
Oh, well,
And
maybe.
it is.
If you can talk to your local, neurologist or neurosurgeon and try to determine if the shunts even needed anymore. Sometimes if shunts are placed for the treatment of leptomeningeal disease and then the pressure dynamics improve, then shunts can sometimes be removed, and then we can have an Ommaya reservoir placed for treatment.
Perfect. Well, thank you so much. This is so interesting. And for everybody on this, we will make sure this information is posted on our website so that you can find a way to contact Dr. Wilcox if you are interested in the study.
Thank
Next,
you.
let's welcome Dr. Forsyth. Dr. Forsyth is Department Chair of Neuro-Oncology at the H. Lee Moffitt Cancer Center and Research Institute and a Professor of Oncological Sciences at the University of South Florida Morsani Medical School. Dr. Forsyth is focused on immunocellular treatments for leptomeningeal disease and brain metastases using various laboratory, translational, and clinical approaches. He has been primary investigator of numerous grants and clinical trials of new drugs and approaches for brain tumor and LMD treatments, and will share some of that work with you today. Welcome, Dr. Forsyth.
Thanks, Elaine. Hi, everybody. Thanks for coming. Just a huge thanks to Elaine for setting up this organization because it's real, I think, a beacon of hope and source of information and support for patients and families when, particularly with this disease, because you don't really know where to turn and to get information you can trust. Just before I go into this, I just wanted to point out a concept, and that is that we sort of always begin our talks with saying leptomeningeal disease is horrible, patients live a few weeks or a few months, and it's incurable. So that last part isn't a true statement, so we have to acknowledge and believe that we can cure this. And let me tell you why that's not unreasonable. It's because there's been hardly any basic research in the area, and all of us, including Dr. Hayden Gephart and several others, believe that you need more science to understand this better, which only makes sense because you can't defeat an enemy if you don't know anything about them, right? You need to have complete understanding to find their vulnerability and what the best way to attack them is. So, we can cure this. It's also not true because patients with leptomeningeal disease from leukemia are commonly cured and lymphoma often cured. So we just need to keep that in mind, that the way people talk about this is really not the way it should be, and it's not the future. And that's partly why we talk about this. So Dr. Wilcox did an amazing job. What we wanted to do is to the bottom right is a picture, believe it or not, of Paul Revere, and his job was really to alert the American forces that the British were invading. And so instead of giving you all the detailed immunology, we're working on making a better Paul Revere. So his job is to, and in this case, he's called the dendritic cell. And his job is really to alert the body, hey, you're being invaded, by bacteria, viruses, or in this case, cancer cells, and you guys really need to get together and fight it. So we wanted to enhance that response. Do I do this slide? Oops. And I think the other thing is about believing you can cure it is you have to believe or you shouldn't be working in the area. Otherwise, you're assuming you're going to fail. So this is based on the work that Dr. Bohr and Wilcox did. And we also observed a similar thing, though they really point out this very well. And that is that there's a whole bunch of immune cells in your spinal fluid, as Dr. Wilcox said, which surprised me because I always thought that they were leptomeningeal disease was just a bunch of watery CSF with a bunch of tumor cells. But most of the cells are immune cells, so it only makes sense that you'd want to help the good ones that are fighting the tumor and hurt the bad ones that were letting the tumor grow. So it's because of this work we decided to focus on the dendritic cell again, who's the Paul Revere. And patients try to fight leptomeningeal disease, where they kind of get some interferon gamma, whatever that is, kind of turn on their Paul Revere's a little, but they can't really kill the tumor effectively. So we wanted to intervene at the Paul Revere stage. So pretty complicated, but immunology is pretty simple because Paul Revere is going to sound the alert. Somebody like Radar from MASH, if you remember that, he's like a helper cell. So he doesn't kill anybody, but he helps people. And of course, we all know the Terminator, those are the T cells that kill everything. And then the goddess of memory, I think what we're doing, it turns on the B cells, which are responsible for memory responses. So when you get a vaccine, it's really a B cell or memory cell function because they make antibodies. So you don't have to fight the same battle. If you didn't have nemocine, if you didn't have memory, you'd be fighting the same battle tomorrow because your immune system would say, oh boy, we're being invaded. What should we do? What should we do? So, in this case, the B cell or nemocine says, no, wait, we already made antibodies. So let's look after it that way. It's kind of and this, maybe I'll skip over, but it's just something in mice where on the left, there's a survival curve for these HER2 mice with leptomeningeal disease. And on the right is a triple negative mouse. And when we give them the treatment that I described, most of the HER2 people, or HER2 mice, are cured, and fewer of the 4T1 or I'm saying triple negative mice. So we took this that was funded by the DoD actually, and we inject these Paul Revere cells, dendritic cells, once a week directly into the Ommaya, which is, I think, the sort of business side of the trial, and then kind of see what happens. Now, the way to collect the dendritic cells is pretty straightforward. It's pretty widely done, but we're basically collecting your immune cells, which is here in the bottom left, just out of your arm or a port. And then we isolate these cells and grow them up in a cocktail. So we're getting all these dendritic cells or Paul Revere cells. We're turning them on with exposing them to proteins that are present on your tumor. And basically, we're giving Paul Revere like 20 cups of coffee and maybe some Ritalin, and really get them jacked up and turned on so they recognize the redcoats as soon as possible. They're ready to go. He's on his horse. He's got his lantern, knows his route. He's ready to go. So it's kind of a non-scientific approach. So this is just the first 6 patients. Now we've treated 10, and we've focused on these because we are doing an in-depth analysis of their spinal fluid because we want to see if our idea that we turn on these cells is true or not. Even with great science and in mice, people are different, right? And so out of these patients, which is kind of the first 6, one person died. She survived whatever 64 weeks is, which is a long time for triple negative, but she died from radiation damage to her brain, which is super sad. And I'll just show you some of those things. Patient 2, for example, is alive and well and doing, I think, really great. It's a little interesting that maybe half of patients, their leptomeningeal disease progresses, but they kind of live with the disease, which is interesting. So you need to figure all this out. And then this is where I need an arrow. I might try, but this may or may not work. No, it's not going to work. Ugh. But on the top left, or the top row, is basically pre-treatment for patient 1. And here we look at all the immune cells and tumor cells that are present. And in the first column where it says cancer cells, before and after treatment, Tracy actually had more tumor cells. So there's more cells in the bottom left than there are on the top right. But she got better. And then the 4th column over where it says, well, B cells, the column where it says B cells, which is the memory cells, there's a lot more of those after treatment. So it looks like maybe that was a correct prediction. And then the column that says T cells before and after treatment, which is top and bottom, there's a whole bunch more T cells, both the helper cells, the radar cells, as well as the Schwarzenegger cells. But more importantly, there's also these memory cells, so that might be useful. So we're trying to figure out how this works exactly in more detail. And speaking of personalized medicine, this is really, it takes months and several $1,000s to do this, but we can actually see what's going on. So this is patient 2, and she has a few in the top left column before and after treatment. Before treatment's on the top, after treatment's on the bottom. She has a few cancer cells, and then they're basically gone, except maybe some of these blue cancer cells. And of course, we don't know what those are, so we need to do more work and take more time and figure out how can we target those. And then the B cell column, there's a lot more memory cells or B cells, and the same story with turning on the T cells. So then there's this other arm that actually Dr. Bohr and Wilcox pointed out in that slide I showed you, that and natural killer cells or NK cells might be important. There's more of those. So I think the point of this is sure I'm good on time. Ugh. So now the next step is in January, where hopefully when we get official notice of funding to start a phase 2 study to add this dendritic cell therapy plus this trastuzumab, or for non-HER2 positive patients, add a PD-1 inhibitor. And then we're going to do all sorts of other fancy stuff to see if we can make what we've observed already better. The second trial to talk about, and I know I have like 2 minutes. The second, and that trial is still open for approval. Now this is combining intrathecal or trastuzumab and pertuzumab, or directly injecting them into the CSF. And this trial is also ongoing and looks pretty promising. Again, on the basis of the science that I probably won't go into too much. HER2 is a single molecule. It's a little, and HER3, those two things have to get together in order to signal. And I'm a little more like HER3 because I sense this ligand in the brain. So the brain makes a protein, neuregulin, that actually turns on HER3. So HER3 gets a signal, but it doesn't have the power to do any work. Whereas HER2 is kind of doesn't respond to things without HER3, but it's the one that does the work. So it's kind of like a little bit like my marriage, where I run around and get excited, but Paula kind of actually does all the work, which might be too honest. But so far, it's very easy to tolerate, and most patients respond. When a single patient decided they didn't want to do it anymore, and that's showing up every week for the first month and then every couple of weeks. And this is just a paper showing that we have stable disease and responses, as well as a patient or two that progress. So that looks pretty promising, actually. And of course, as usual, what we want to do is just 2 more slides. What we want to do is, since we know something's working, what this clearly is, that we want to make it better. So we have a pretty big team that's interested in leptomeningeal disease, by the way, because they know it's a desperate disease about which little is known. And, it's because of the patients and families that they trust us and support us and allow us to collect tissue, by the way. So the first trial that I talked about with the revered dendritic cells, we have a couple slots that are open. This intrathecal Herceptin progetta is open and accruing. I think there's about 9 more slots. And then as soon as you get official notice of funding, I can put down the phase 2 that would be expected to start in January. So talked for 13 minutes. I'm sorry.
Nope, you did great. Thank you. That was perfect. And I love your positive way of opening this. We do need more research and we need to talk about this with more hope. And as you said to me when we met, science is hope. So I always appreciate that because I think that is the truth. We just don't know enough. So we just need to get deeper into this research. We did have a few questions. So one thing I know you've talked about HER positive. Are HER2 low patients eligible for your trial?
Yeah, not so far. We haven't included them yet. We may or may not go on to do that. We're in the process of submitting it to all the regulatory bodies, so we're going to wait till it gets through the DoD, and then we'll probably make one last change to include that. We do need
Okay.
to get, and of course, we do need to get some data too.
Yeah. And what about any phase 1 patients? Can they join phase 2?
Yes, they can join the phase 2.
Okay, great. And then someone asked about patients who have HER2 positive breast cancer just seem to have a longer prognosis that they feel like when they've looked at the LMD
Yeah.
research. Do we know why?
No, that's a great. No, I don't. I don't know why. That's a great question. I mean, unfortunately, I'm prepared to answer the other question, is why it loves to go to the brain and the cerebellum in particular. Is because the neuroregulin protein is there. I mean, the answer must be that the body is just better at fighting it, and we can figure that out too,
Mhm.
because the pathways that look after how Herceptin and Perjeta are described, like it uses complement and other things, so we can figure that out. That's a great question. Oh, never mind. Okay. I mean,
Um,
the thing with her too is interesting because it's actually a little immunosuppressive, which is kind of interesting, like biologically. So we need to figure out how those two things are connected.
Great. Yeah, that I think it's a really interesting part. And I know that we've talked about that a lot of trying to figure out why some people are responders, why some aren't, why some cancers seem to respond better, why some don't. So that's part of the research we need to do. I
And
do know that
I
we've
think,
got,
sorry, and I think with the single cell, that's kind of what we want to do in real time is, figure out before you treat somebody, oh, look, it's not going to work because you have too many whatever cells, or, oh yeah, you really need to try this,
Mhm.
as well as, there's no reason why we can't detect recurrence earlier. There's no scientific reason. Should be surveilling people so you can jump in quickly
Yeah,
rather than waiting till somebody winds up in an emergency.
absolutely. We do have a question about that these studies seem to be for breast cancer LMD, and are there equivalent ones for EGFR positive non-small cell lung cancer? But I do know that Dr. Hayden Gephart is going to be speaking about
Right.
that. So thank you, Dr. Forsyth. And we are going to go to our next speaker. We have Melanie Hayden Gephart. Dr. Hayden Gephart is a neurosurgeon treating patients with malignant and benign tumors. She directs the Stanford Brain Tumor Center and the Stanford Brain Metastasis Consortium. Her lab seeks greater understanding of the mechanisms driving tumorigenesis and disease progression in malignant brain tumors. They develop novel cerebrospinal fluid-based biomarkers to track treatment response, relapse, and neurotoxicity. Their bedside to bench to bedside research model builds on a foundation of generously donated patient samples where they test mechanisms of brain cancer growth, develop preclinical models, and facilitate clinical trials like the one she will discuss today. Welcome, Dr. Hayden Gephart.
Thank you so much again for having me. And it's such a privilege to follow these other esteemed speakers who also have done such a great job of setting up some components and considerations of the trial that I'm currently running. So just to start, greetings from Stanford. Somehow, Helene, you've been able to gather all corners of the country, but I do really want to reinforce how important it is to have a good working relationship with your local team, and that if and when there's a trial that's worth traveling for, your team will let you know. And of course, we do the same for our patients. In disclosures of this data that I'm presenting, some of it comes from my lab in terms of the preclinical data, and then also the phase 2 trial that's currently ongoing has a component of it expanding from breast cancer to other patients with other primary tumors and leptomeningeal disease that's supported by the company. Just a bit about me, as was mentioned, we all have different backgrounds and expertise, but I'm a brain tumor neurosurgeon, and my laboratory has a basic science component, as was already mentioned, trying to come up with critical insights as to how the cancer cells actually seed the leptomeninges in the first place, and once they're there, how best to do it. It's a privilege to work with my patients. This is just an example of a thank you card that a patient hand stitched for me, and really that interface between coming up with new treatments and providing the best options for today is really critical. So just to start again, as a brain tumor surgeon, brain metastases are the most common malignant tumor that we see in the brain. Now, leptomeningeal disease in and of itself is not operative in the sense that we're not going in and surgically removing the cells, but we do provide a service of creating a port, so a ventricular access device, commonly also known as an OMIA, to sample cerebral spinal fluid. But we also know that the incidence is increasing and patients are frequently excluded from clinical trials. The samples that are obtained are rare to get and difficult to acquire. And so it's critical for all of us to share. And everyone who's here, of course, we work together nationally to try to share our data and come up with better tools. Here's just an example of a very large tumor depicted in purple that sits between the motor fibers in dark blue and the sensory fibers in light blue, as well as next to the ventricle, the fluid pocket in the brain. And one of the difficult things is that even when the tumor here is resected, a significant number of patients ultimately will go on to develop leptomeningeal disease. Most patients who do have not had surgery, but this is a classic MRI scan, T1 with contrast, axial and sagittal MRI scan showing the typical way that the cancer cells actually grow along the folds and the peaks and valleys of the brain. And as you see this, of course, you can understand too why this is not considered an operative disease. And yet it's something that I've really centered my laboratory and my clinical focus on and new clinical trials because it's such a challenging condition for the patients that I do treat. And what we realized long ago is it possible, this is actually over more than 12 years now, that if we can identify them soon enough, again, with metastatic disease across many different tumor types, and the patients are stable, then if they could get radiosurgery to the tumor before surgery, then that potentially could decrease their risk of developing leptomeningeals afterwards. And so when we look at the incidence of leptomeningeal disease, either with or without surgery, certainly post-operatively, the risk, if the tumor is in the back part of the brain and the cerebellum, the risk can be up to 30%. And so what teams at our place and others started to do was actually treat patients, presents with a symptomatic tumor that clearly needs surgery. But if we're able to wait just long enough for the patient to get radiation first, then that should potentially decrease the risk. So here's a QR code and link to a currently ongoing clinical trial, which is open at Stanford and several other facilities, that's actually randomizing patients into getting radiation either before or after surgery. Radiation after surgery to the cavity of where the tumor is the most common current standard of care, but it's possible that doing the radiation first in carefully selected patients could help to decrease the incidence of leptomeningeal disease. So that said, though, of course, I see many patients who never had surgery and already have leptomeningeal disease. And to just present the challenge that I was looking at, again, many years now, a company had come to us because we had developed a new model of breast to brain leptomeningeal disease where the mice did not have systemic disease. And they had developed a new molecule that targets the LAT1 transporter. So LAT1 is a very well-known transporter. As probably everyone here on this call knows, it's very challenging to get drugs into the brain, even if they have great systemic treatment effect. And part of what made this molecule interesting was that it used this LAT1 transporter, which is a transporter that's used to get amino acids into the brain. It's also been used previously for treatment of Parkinson's disease and for gabapentin or Neurontin to treat seizures. And this molecule was combined with something that recognizes LAT1, which also happens to be very highly expressed in tumor cells across multiple tumor types. Again, I'm speaking here about breast, but it happens across many primary tumor types. And the drug can get in and then selectively bind to the cancer cells, and it has a cytotoxic component that then causes DNA damage that the tumor cannot repair. And the fortunate thing was that we had developed a model in the lab to be able to carefully and quickly test this to show some indication of efficacy in order to allow us to move on to a phase 1 clinical trial. So these mice, just as in humans who develop this classic pattern of the cancer cells covering the surface of the brain and the spinal cord, the same thing happens in mice. These blue arrows here depict both live imaging of the mice who have cancer cells only in the brain and the spinal cord. And we can see on the pink images, those are pictures that show the cancer cells in the darker color, again, coating the surface of the brain and spinal cord and the small nerve rootlets. In this case, the drug, whether we gave it in the belly or through the vein, showed an improvement in survival, decreased growth in the tumor, and it was relatively well tolerated in terms of toxicity. So the phase 1 went on to determine the dose, but one of the things that was really critical when I was designing the study is that I needed more information about if and when patients did not respond to the drug, why. And so we see the drug is given every month, and all patients have to have a ventricular access device, so again, a reservoir, as it's been described, just like a port that patients use to get chemotherapy to put drugs into the heart, but a small tube that goes into the fluid-filled pocket of the brain that's connected to a subcutaneous reservoir. So a small little pocket under the skin that can be readily accessed to draw fluid and provide medication. And in this case, the patients are seen every week for routine lab draws, but then also to have the cerebrospinal fluid tested. And so that allows us to develop new biomarkers. And one of the things that we've worked very hard on is to come up with new ways to look at CSF. Historically, just looking at the individual cancer cell underneath the microscope provides very little information about what that tumor cell is doing, how it's potentially interacting with normal cells or brain cells. And so we've developed new ways to look at components that are not even necessarily part of the cell itself, but can give us indications about brain health and disease, and also response and resistance to therapy. So one of the considerations that we need to keep in mind when it comes to clinical trials is that these trials are under very high demand. Part of that is that there are very few trials that are available, and so we need to consider how much research support. I also feel very strongly that patients should not have to travel extensive distances for a trial for which then they'll not be considered eligible. And so a lot of that comes with screening patients ahead of time in order to relieve a lot of those obstacles. The screening has to be quick. Patients, when they become symptomatic, tend to have those symptoms progress relatively quickly. And so very rapidly coming together, everyone on my team has my cell phone. We quickly try to figure out where the best trial and options would be for the patient. If the patient needs a reservoir, then of course I add them onto the surgical schedule as quickly as we can. One of the issues with sort of target-based therapy is, as someone asked previously, we didn't really think up until recently that patients who were HER2 low would respond to HER2 targeted therapy. And it just shows that in part, we don't always understand how a drug will be successful. And also the challenge in determining target expression, particularly for patients like in leptomeningeal disease, where there's no indicated biopsy. In our trial, what we decided to do was look for any type of metastatic tissue that the patient would have had sampled previously, and not wait for enrollment, but then consider that when thinking about overall response to the drug. again, having access to CSF is standard of care for patients with leptomeningeal disease, allows for ready CSF access and for biomarker development. Here's an example of ways to get in contact with us. Again, it's very difficult on clinicaltrials.gov or on other websites to keep them up to date because the slots for a trial, the eligibility, whether it's a good fit for you is probably best determined by your local team. But we have a direct phone number that patients can call and get a second opinion and talk to the trial team to see if that could be an option for you. The most important thing is we don't want you to feel like you're on an island alone, that this is really a growing network of physicians and scientists and patient advocates all coming together to make sure that people understand what treatment options are available and how we can best facilitate care. I feel very fortunate. This is just an example of a patient message sent to me in Epic to be able to provide these opportunities for patients. But of course, this wouldn't be possible without the scientific community and also the incredible generosity of patients to donate samples with the clear understanding that it may not be of benefit to them, but it benefits scientific progress overall. And so with that, I want to thank the sources of funding that have allowed this work to continue, my laboratory and my clinical team, and I'm happy to take any questions.
Thank you, Dr. Hayden Gephart. That was excellent. We have a question about proceeding with RNA sequencing and how you extract the RNA. At times in CSF, depending on the patients, the amount of material is very low, not allowing further analysis.
Yeah, that's a very good question. So there's two main approaches. One is based on trying to capture the individual cells, and that can be challenging because the number of cells that are actually in the fluid are few. We feel fortunate because we've been able to, for messenger RNA, actually be able to capture the messenger RNA, the actual signal kind of in the cookbook that's getting sent to actually do the cancer and normal brain cell processes that are important both for function and for disease progression. And that we've been able to actually extract and sequence in an unbiased, meaning sort of whole transcriptome, all of the mRNA that we can see way, rather than having a panel where you have to have a preconceived notion of what's important. And fortunately, that's been successful across many different disease types and primary tumor types. And those data and all of the methods will be freely available to the scientific community, hopefully by the end of the calendar year.
Excellent, thank you. This is relevant to your trial, but also probably Dr. Wilcox's, but there's a lot of questions about trials appropriate for someone with spread from non-small cell lung cancer, renal cell carcinoma, non-Hodgkin's. Are you taking patients with these various tumor types and cancers, and are you seeing any results?
Yeah, so one of the challenges that we saw early on was restricting the drug too much, particularly in a drug target that scientifically should be expressed on most metastatic cancer cells. And so we quickly expanded it to all subtypes of breast cancer. And then based on promising data in the first 3 of 10 patients who received the drug who had extended survivals or radiographic response to the drug alone, we did expand a third cohort, which was leptomeningeal disease, all comers. Something that I'm sure Dr. Wilcox could speak to as well. But one of the challenges with targeted based therapy that is true, say, for patients who have melanoma or thyroid cancer, lung cancer, is that it is very difficult to request that the patients be washed out of their systemic drug. So unlike one of Dr. Wilcox's trial that requires stable systemic disease, in our trial, it's delivered IV, and we would expect that patients should respond in the brain and systemically if it's going to be helpful. However, the downside is that it does require a washout of the systemic medication because there's many different types of drugs and we don't know the individual reaction. That's much harder in EGFR mutant lung cancer, where even though there's progression on the drug, it's still probably doing something. So those are details that we definitely need to work out with trials moving forward, is how to approach washout windows.
Thank you. That's perfect. I'd like to bring the others back on now. So if we can answer a few of those other questions, I want to just check with Dr. Wilcox if she had any thoughts on the last answer that Dr. Hayden Gephart answered.
Yeah, of course. Thank you. So, we also ran into a similar problem of if you restrict access too much with too, restrictive uh, primary cancers that are allowed to enroll, then you're never going to be able to accrue, this to your study in a timely fashion. And you really want to give access to this drug to as many people as possible. So for the deferoxamine study, we allow any solid tumor. So breast, lung, renal, prostate, what have you. It's restricted to solid tumors, not lymphomas or leukemias, primarily because that is what was tested in our preclinical mouse experiments. But that's not to say that there might not be, open to hematologic malignancies like lymphoma and leukemia sometime in the future. But for right now, it remains restricted to solid tumors. And I'll also say that um, we don't really have too much in the way of washout periods for enrollment on the study. Patients can enroll whether they have, stable extracranial body disease or progressive extracranial body disease. We expect that our patients are going to have to receive systemic, intravenous or oral cancer-directed therapy to take care of everything below the neck. And so, we've built into our eligibility criteria that if they have to, change to a different line of systemic therapy for the rest of their body, they can start our drug, the deferoxamine, after 3 weeks have passed, just so that we can confirm that the cancer is still there in the spinal fluid and that they're tolerating the other drugs well. But if they're on, for example, Tagrisso for lung cancer, and the Tagrisso is keeping everything in the body under control, but there's progression in the leptomeninges, then they can stay on the Tagrisso and then just start with the deferoxamine. So we try to make it as simple as possible for patients to enroll.
That's helpful. I know, Dr. Hayden Gephart, you mentioned travel, and we do get a lot of questions of people in Europe, Australia, and saying if, someone lives nowhere near there, is there anything that can be done? If they're willing to travel, is that okay? I do want to just say that we are going to be having a webinar with some of the European specialists who can talk about trials going on there, but just if, is there anything you can tell them? Because I know that Australia is always a question for people of what to do.
Yeah, I'm happy to address that. I suppose I'm sensitive a bit to the hardships of travel, the financial burden that can create, as well as the time and time away from family, time away from work. And so I think if there were a clinical trial that I was really excited about or that I thought that would be a great fit for the patient, then I absolutely would have no reservations about referring the patient to New York or someplace that that I thought would be a good fit. The challenge is, I think that it's very difficult for patients to be able to review all of the inclusion and exclusion criteria, which even me as the leader of my own trial, I have a whole team of people who are helping me to ensure that a patient is eligible. And so I would encourage to look out for it. And if there's a capacity to even get kind of a remote second opinion, that can be really helpful just to gauge the field. Because sometimes what I found is maybe patients are interested in coming on to my trial, but it turns out that maybe we have a better option that doesn't necessarily even have to be given on trial or can be facilitated with the patient's local team. So I think that communication and keeping those lines of communication open, we're always happy to contribute to that.
Well, thank you so much. We are out of time. This was really great. We just had so many questions. We couldn't get to everything, but I do feel like we covered a lot. So I truly hope that this was helpful for everybody on this webinar and that you're leaving today feeling more informed. Thank you all so much. I want to say thank you to our incredible panelists, our amazing producer, and to all of you, our audience members. And just be sure to follow us on social media, sign up for our newsletter on our website, lmcancer.org, so you can stay up to date on all of our upcoming events. Thank you again, Dr. Hayden Gephart, Dr. Wilcox, Heather, and Dr. Forsyth for everything. We really appreciate all the work you're doing, all the research, and we hope to see you again soon. Have a great day.